temporal resolution. So for this system, the temporal resolution is the good, more than 17 per second. Based on this system, we use this system can able to perform rapid and efficient analysis of clinical tissues with multi-parameter information excited. We achieved a pathological diagnosis accuracy, and for the as we know, the HE is the gold characterization for the clinical detection, and our imaging is similarity of higher than the 19% compared to the HE imaging. Here, I will give 3 samples of our works about the clinical tissue image analysis based on our system. The first one is the clinical tissue, our cancer tissue imaging, and we use our system to get the clinical ovarian cancer tissue, get the multimodal imaging for, based on our system. So for ovarian cancer tissue imaging, we can analyze the collagen protease by SG imaging. And in addition to SCG, we can use the SS to get the lipid and the proteins analysis. So we can perform the quantitative analysis of the distribution of the lipid and the protein molecules. And as well, we can get the intention and the peaks difference of the lipid and the protein molecules in SS spectra. The second is we use our system to do the clinical skin cancer tissue imaging at the same. I mean, so we use the same, including we use the same way to get the multimodal imaging from the clinical skin cancer tissue based on the multimodal system. And also, we got the multimodal nonlinear optical imaging from the clinical glioma. From the information, we can know that the lifetime of the NADH and the FAD of clinical tissue are different at the different stage. So based on our system, I means we can make the analysis of the clinical tissues. We can get the different information, and we can analyze the different stage of the cancer. Okay. Furthermore, we proposed a nonlinear optical signal enhancement method to improve the imaging quality of our multi-parameter optical microscopy, and the SNR and the resolution we can highly improve after the network learning. We use the deep learning to improve our our imaging, and increasing the capability of our system for the fast diagnosis. So for nonlinear optical imaging, that still have some challenge when the standard deviation of noise is similar to the imaging signal, and we can use the denoising model to get the enhancement of the imaging resolution. And our method can avoid the burning and clearly analyze the detailed, such as we can get the red blood cell from the tissue, and we can get the collagen fiber imaging from the tissue very clearly. And so we use this way can greatly improve the contrast and can enhance the speed of the imaging, also can enhance the resolution. After the network, we can get the more clearly imaging, and they can do the more deeply analysis of the clinical diagnosis. So for the first amount, the unmarked nonlinear optical microscopy can provide the rich biomolecular information for environmental analysis of the cancer hint generis. Hint generis is very popular for the tumor microenvironment. So, however, it's difficult to get the microstructure from the tissue. So for our so we developed this deep learning method. Based on this method for this technology, maybe we can enhance the imaging, because the for nonlinear optical imaging has a low clinical imaging, and maybe compared to the other imaging method, they can give the more information, but they are still have the low the signals. And for HE still have a need to the for the HE is a traditional staining method, and need the require a longer preparation process and need more times. So we think if we can use the deep learning to restructure the imaging like the HE, is very conference for the doctors. So therefore, we developed a nonlinear digital histopathologic through the neural networks, and we can transforming unlabeled nonlinear optical imaging of clinical tissues and into the digital his pathological imaging, and we need without the need for the long screening process. That means we can get the digital pathological imaging in the short time. We don't need the more time to prepare the samples. Here is we proposed the method. We use with this new method for constructing the base for unlabeled the tissue imaging. We use the differential activation method to perform the differential operations on each staining images and the activation layers to significantly highlight the difference between their nuclear and hot plants, and obtain the distribution of the nuclear. So we can find the very clearly the cell imaging from the tissue. Uh, for this method, we get this digital pathological imaging can also be used for the nuclear segmentation. We demonstrated this label-free segmentation of clinical organ to get the clinical organ cancer tissues. Our deep learning approach can clearly distinguish the cells and have a higher contrast than the HE imaging. Well, this method completes the structural information and also can obtain it from the nonlinear imaging, and quantifies the distribution of cancer cells in pathological tissues. And so we can get the more information. We can get the more clearly the image cell imaging here, and compared to the HE, so we can get the more clearly after the neural networks. Based on above works, there still have some challenges to do. On the basis of the two-dimensional information denoting models, we further developed a three-dimensional dimensional imaging denoting method. We called the three-dimensional self-supervised optical signal enhancement method. Based on this method, we can achieve denoting of three-dimensional imaging frequencies without we don't need to collect the high signal to noise rote images. For this three-dimensional imaging, we can get the clearly imaging and use this method. I mean, we use this. The network can achieve high reconstruction performance with the field delay test, and without the need for the additional large-scale training device. Here we simulated the neuronal signal on the two-photo microscopic imaging and to get the calcium imaging data. As well, we can get the dynamic imaging of the environment of the calcium from the brain, mouse brain, and due to the influence of the short noise and the signal to noise rate of the imaging data is very low, and we can get the imaging with very clearly. Also, we compared our method with the previously reported method, and the stronger the neuronal distransition. So the report method can present the blurry reconstruction, and for our method, we can get the cancer imaging in data. So based on our system, we can get the very clearly image, and also compared to the input data, the 3D signal has improved by more than 9 times, and we can decrease the noising, which is the very is 4 times higher than the original data. And also, okay, here is our works for the nonlinear optics, and we use our multimodal nonlinear optical microscopy to get the imaging, and with the deep learning to improve the resolution and improve the speed. And so now let me shift to the final part. For the afterline, I think we also to develop of our techniques, including the nonlinear optics, and we want to do the in vivo imaging, like the brain imaging, and to reveal the relation of the optical signals and the pathological with the high speed and the high resolution. And now I will finish my talks, and thanks for your attention, and thanks for my collaboration and my students, and so on.
Thank you, Liwei. Thank you very much for the very impressive results. So let us thank the speaker. And now the session is open for discussion. So please, questions? Questions from the audience? Okay, so actually I have a couple of questions. So my first question is, are all of your application ex vivo, or do you have some in vivo application of your technique?
So in vivo, we do the cell imaging, tissue imaging, and in vitro we will do the mouse brain imaging.
Okay.
In future, we will do the yeah.
Great. And my next question is, what is the typical time to get the answer for your technique? So you mentioned that it's quite fast. So what is the typical time which is required to get the results of diagnostics?
For this part of the front, it means we use the deep learning to how to difficult for the enhance the speed.
And what is the time in seconds or in minutes? So how long does it take?
Sorry.
So what is the time for analysis of one sample? What is the time? How long does it take with your technique?
Oh, more than if for the tissue imaging, more than 1 or 2 minutes. Use our multimodal this system.
Okay, great. Thank you. So any other questions from the
Thank you.
audience? Okay, let us thank the speaker again. Thank you very much, Liwei, for very impressive results. And so now,
Thank you.
thank you. And now it is my big pleasure to give floor to Dr. Zhi Li from Zhejiang University, also China. So
Yeah. Hello everyone. So could
hello.
you see my report?
Yes, everything is perfect. So
Okay.
the floor is yours, please.
Okay. Thank you very much. Thank you for the introduction. And hello everyone. My name is Zhi Liu. I'm from Zhejiang University, China. And the topic of my talk is multiparametric high-resolution imaging of biological tissues relying on endogenous contrast. Okay, so first of all, let's take a look at the background. So as we all know that fiber-like structure is one of the most prevalent structures in organisms. So there are lots of fiber-like structures, including such as the blood vessel, the collagen fibers, the neural axons, the cytoskeleton, and so on. And the remodeling of fiber-like structures is closely associated with the progression of different diseases, such as the diabetes, the cancer, the neurodegenerative disorders, the cardiovascular diseases, and so on. For example, the progression of cancer accompanies the cell matrix interactions, and these interactions can lead to collagen fiber remodeling. Thus, if we are able to quantify the subtle remodeling of collagen fibers, we would gain a better understanding of cancer progression. Okay, so in this context, we are trying to acquire the images of fiber-like structures in our group, and we developed some optical metrics for the quantification of these fiber-like structures. So, although the three-dimensional fiber-like structural images are more and more available, unfortunately, current analysis of fiber-like tissues still mainly relies on the analysis of 2D images. In this context, we have developed algorithms which are able to acquire voxel-wise three-dimensional orientations of fiber-like structures in biological tissues, which shows better accuracy and time efficiency than previous efforts. And then, based on the three-dimensional orientation information, we have developed several optical metrics which are able to quantify the morphology and organization of fiber-like structures. So the first one is about the fiber alignment, and we call this as the 3D directional variance. So 3D directional variance ranges between 0 and 1, with 0 corresponding to perfectly parallel alignment, while 1 corresponding to complete randomness. And as we can see from these represented images, there are two regions. One is with parallel fibers, and the other is with completely random fibers, and they show very different 3D directional variance levels. Okay, then the second metric is a metric of morphology, and it is called waviness. So waviness is a measure of orientation variation, which quantifies the fiber crimping level. And again, this waviness metric is also a normalized metric, which ranges between 0 and 1. The higher the waviness value is, the more cramping the fibers are. Okay, and then the next metric is organizational metric, which is the local coverage. So local coverage assesses the collagen content at a localized region, in contrast to the previous overall density, which focuses more on the average content level at a given field. And this local coverage metric also ranges between 0 and 1. The higher the local coverage value is, the denser the fibers are. Okay, finally, we developed the thickness quantity, which is able to provide a direct 2D or 3D visualization of fiber diameter. It is a completely automatic metric without any manual determination of parameters, and it shows very good performance under different fiber density and alignment conditions. So after developing these optical metrics, we have applied them into the biomedical applications to gain a better understanding of lots of important diseases. So the first one is about the pancreatic cancer. So here I show in the left, I show some representative 2D and 3D images of pancreatic cancer tissues and the normal controls. So here we use the two-photon excited fluorescence to obtain the images from the cell clusters, the blood vessels, and the elastin fibers. And we use the second harmonic generation to acquire the images of collagen fibers. And then we use the collagen fiber images only for the subsequent assessment of the three-dimensional orientation and organization of collagen fibers. Okay, so here I show the representative distribution of three-dimensional orientations. As we can see from the representative maps and distributions, fibers are cross-hatched in the normal tissues, while the curvy features disappear in the tumor specimens. And there is only one single peak of theta at about 150 in tumor tissues, in contrast to a wider, possibly bimodal distribution in healthy samples. Based on the three-dimensional orientations, we then acquired the 3D directional variance, which quantifies the alignment of collagen fibers within the pancreatic tumors and healthy tissues. As we can see that the tumors shows the collagen fibers in tumors show much better alignment than that in healthy tissues. So a possible explanation is that there are lots of newly synthesized collagen fibers in tumors during the progression of cancer, and these newly synthesized collagen fibers are very well aligned. Okay, so besides the alignment, we also quantify the cramping level of these collagen fibers, use the waviness metric. And here, from these representative maps, we could say that fibers are wavier within the normal tissues, as what we mentioned above. Therefore, we could say that waviness measure also provides supplementary insights into the morphology of collagen fibers in pancreatic cancer. Okay, and then we combined the complementary information from different angles, including the 3D directional variance, the alignment, the waviness, and the collagen fiber content for classification of cancer tissues from the normal ones. And finally, we obtained a very high sensitivity and specificity in distinguishing the cancer samples. Okay, so the next application is in the ovarian cancer. So as we know that ovarian cancer has the highest mortality rate among all gynecological cancers. And here we mainly use the local density metric to quantifying the morphologically localized distribution of collagen fibers. And we assessed three types of tissues: the normal one, the benign tissue, and the malignant human ovarian biopsies. And as we can see from these representative maps, different tissue types exhibit obvious differences in the local coverage and its depth-dependent features. Okay, so finally, by using the fiber density related metrics, we achieved an overall diagnosis accuracy of 96.3% in distinguishing the normal, benign, and malignant tissues. Okay. And then the next application is the hypertrophic scar. So here we try to combine all of these optical metrics. So we know that the hypertrophic scar is a aberrant form of wound healing process. However, its clinical efficiency is limited by a lack of understanding of its pathophysiology. So here we use the two-photon excited fluorescence to acquire the endogenous images of elastin fibers, and we use the second harmonic generation to acquire the images of collagen fibers. And then we used the multi-fiber matrix analysis model to map the structural remodeling of the ECM in hypertrophic scar. So here I show some representative maps of multi-fiber matrix, including the 3D orientation, directional variance, waviness, and local coverage from both the normal tissues and the scar tissues. Okay, so it is very interesting that we also perform this analysis to try to identify the scar boundaries. And by using a combination of all of these metrics, we could successfully identify all the boundary tissues. So it makes good sense in clinic because identifying the boundaries can help the doctors to fix the extent of surgery. So it has some translational potential in this context. Okay, so finally, we applied this imaging and analysis to the study of port wine stain, short as PWS. So here we do the in vivo assessment of three-dimensional microstructure of blood vessels in PWS. So PWS is a congenital cutaneous vessel malformation. However, the microstructure of these vessels still remains largely unknown, and here we use the OCTA method for the acquisition of vessel images. So OCTA is another label-free method for the vessel imaging. And again, we applied our optical metrics to assess the three-dimensional morphological and organizational features of blood vessels. Okay, so as we can see that the blood vessels within the normal tissues and the PWS tissues are very different. Specifically, we could see that blood vessels within PWS tissues are much thicker, more disorganized, and denser. And also, it is very interesting to note that some features, such as the 3D directional variance, can only be resolved by the 3D analysis. It highlights the sensitivity of 3D analysis versus 2D analysis. Okay, so and then by combining these optical matrix, we achieved a nanosafe value of 0.98 from the ROC curve, indicating that the blood vessel signatures can serve as a very good tool for the identification of PWS. Okay, finally, it is the summary of this talk. So here are lots of typical fiber-like tissues in the organisms, such as the collagen fibers, the elastin fibers, and the blood vessels. And these tissues can be imaged via completely endogenous contrast. And in our group, we have developed several representative optical metrics to quantify the morphology and the organization of these fiber-like tissues, such as the three-dimensional orientations, alignment, waviness, and local coverage and thickness. And these optical metrics can provide complementary insights into the remodeling of these fiber-like tissues, which ultimately can lead to a better understanding of some important diseases. Okay, so that's all for my topic, and many thanks for your attention. I'm happy to take any questions. Thank you.
Thank you so much, Dr. Liu.
Thank you.
Thank you for the very
Yes,
impressive
yes.
talk. And now we have some questions from the audience.
Yes.
Thank you very much for a very nice presentation. Well, you made a great success. The question is, are all these
Yes.
approaches and modalities are introduced into clinic already, or you are just in the process of approbation and experiments?
Okay.
Thank you.
Thank you for your question. So for now, it is still. I mean, not. We have not yet do the medical translation yet. It's still on the stage of the research, but we are trying to push them and translate them. I mean, maybe in near future. Yes. Thank you.
Okay, thank you. We have several more questions. So, Dr.
Okay.
Thank you very much for the very interesting
Thank you.
presentation. And my question is about the visualization of vessels that you demonstrate
Yes.
At the end of your presentation. What was the depth
Yes.
of the signals that you registered for visualization of the blood
Okay. So
vessels
okay.
in
Yes.
normal and in pathological case? Is
Okay.
there
So
any
you mean the
differences in
depths of the imaging,
pathological?
right?
Oh, okay.
Yeah. So do you mean the depths of the imaging? So here we use the OCTA, and for OCTA, for this optical method, the typical depth of imaging is in the millimeter level. So it's about 1 millimeter or so, I think.
And what was the type of the tissue that you used for these experiments?
Oh, this is the okay. So this is the skin on the face. So here is the schematic. So this is skin of the face of the face of human beings, and we do the in vivo imaging from I mean we do the in vivo imaging of the skin face skin,
I see.
and collect these images from the hospital. Yes.
Thank you very much.
Thank you.
We have more questions.
Thank you very much for
Yes.
your presentation. I have a question. Can you use your
Yes.
approach of data analysis to make mapping of the filaments inside the cells? Like, not in tissue, but inside
Okay.
the cells.
Yeah, it's a very good question. Actually, yes, we definitely actually we could use this optical metrics for any fiber-like images, such as, for example, we have published a paper in Optics Letters we quantify the remodeling of the ER and the microtubule. So they are very typical subcellular cytoskeleton and organelle. So definitely, yes, we could apply our metrics into the application of the cell images. Yes.
Oh, thank you much. That sounds great. And what is the requirements, signal to noise ratio or the fluorescence intensity? What are the requirements for a good contrast of your map?
Yes. So okay, it's a very good question. However, by now we have, I think our algorithm has a very good tolerance to the image quality. So we have not yet tested the image quality dependence of the analysis, but by now they have been successfully applied to different types of fiber-like images and achieved some reasonable results. Okay,
Thank you very much. Thank you.
thank you.
So actually, I have a lot of questions to you, but unfortunately our time is limited. But maybe I can ask a couple of them. So the first, my
Yes.
question is, if have you also tried to evaluate the average length of fibers in your analysis? Because I presume that it could provide some
Yes.
additional diagnostic quality. So have you considered this
Yes.
kind of
Okay.
parameter?
Yes, it's a very good question. So unfortunately, we haven't developed the quantity of lens in our group. But I think here are some efforts which have already focused on the length of the fibers. But in our group, not yet.
Because I think if you add this information to the orientation
Yes.
information, this could provide some
Yes,
additional benefit.
definitely. It's a very good quantity. Yes, it
Uh,
can be very useful in lots of applications. Yes,
so this is great. And actually, my second question is, if
yes.
have you tried more differential diagnostics, for example, to study how the parameters depend on the cancer development stage? So it's can you start? It's just early
Oh,
stage of cancer or late stage of cancer. Have you tried this
yes,
kind
yes.
of
It's a
analysis?
good question. So yes, we will publish a paper there. We use this metrics to stage different to stage the cervical cancer. So the early stage, the late stage, and the pre-cancer stage. So yes, we are working on that.
Great, great. This is really great
Yes.
to hear
Thank
that.
you.
So thank you very much. We don't
Thank
have any
you.
questions, so thank you again. And let us thank both
Thank you.
speakers because we are closing this session. Thank you for all.
Thank you.
Yeah, and it is my pleasure to close this session. And now we have some coffee break, so please join us after the coffee break. Thank you.
Thank you.
Hello for everyone again. We continue our Russian-Chinese seminar, and first speaker, Dr. Mikhail Kirillin. He will present the talk with the name Analytical and Numerical Models for Advancement of Diffuse Spectroscopic Techniques. Mikhail is from Institute of Applied Physics of Russian Academy of Science. Please, Mikhail, give your invited talk.
So, thank you, Andrei, for the presentation. And first of all, I would like to thank the chairs of the biomedical diagnostics session, namely Professor Priezhev and Professor Tuchin, who invited me to give a talk here. And today I will tell you about our modeling results in the field of diffuse optical spectroscopy. So unfortunately there is a mistake in the program, but if you are interested in dual wavelength monitoring of PDT, I can give you a brief summary later on. Oops, doesn't work. Okay, let's try in this way. So I will start, so this is the outline of my talk. I will start with the motivation, and then my talk is divided in 3 parts. So first of all, I will start with some results of Monte Carlo simulations, which allowed us to generate maps of diffuse optical spectroscopy signals. Then I will discuss a little bit about the probing depth. So Monte Carlo is a technique which allows us to analyze the probing depths of the technique at different wavelengths, and I will discuss the effect of this depth on the results of reconstruction of saturation in this technique. And in the final part of my talk, I will tell you about our novel analytical model of DOS signal. So the motivation, of course, the main motivation is active employment of spectral techniques, which have actually already been introduced into clinical practice. And I will focus on diffuse optical spectroscopy and in more details on wideband diffuse optical spectroscopy. The physical feature I would like to tell you about is also strong dispersion of bi-tissue optical properties in visible and near-infrared ranges, and its effect on formation of on diffuse optical spectroscopy signals. And of course, the general motivation for all our studies in the field of biophotonics is our trend towards personalized medicine, and of course, novel diagnostic techniques will allow us to improve the treatment protocols. And in my talk, I will focus on analytical and numerical models of diffuse optical spectroscopy signals, and also on the choice of spectral range for reconstruction of different tissue parameters. So I hope most of you are familiar with principles of diffuse optical spectroscopy. So I will concentrate on the source detector modality when there is source or several sources and several detectors. And actually the distance between source and detector can allow us to tune the probing depths of the technique, and so different source detector separation provide us information from different depths. So, and the basic aim of application of diffuse optical spectroscopy based on diffuse reflection spectral reconstruction of the content of the main tissue chromophores with quite well-known absorption spectra, which are shown here, and the main chromophores are fat, water, oxy, and desoxyhemoglobin, and melanin. In our laboratory, we are developing a so-called self-calibrating probe approach, which employs 2 sources and 2 detectors with 2 source detector separations, which allow for fast reconstruction using classical diffuse approximation of radiative transfer equation. And employment of 2 pairs of sources and detectors allow us to avoid distortions in the optical measuring tract in the fiber system. And actually, Dr. Ilya Turshin have already gave a detailed presentation earlier on this topic. So we are using this quite short formula to extract effective attenuation coefficient from which we can extract the absorption spectra and reconstruct the content of different chromophores in tissue. So I hope most of you know the principles of Monte Carlo simulation. This is a statistical method which is based on numerous simulation of random photon trajectories. So we can efficiently apply it for simulation of those signals. And in here we employed massive simulations to build two-dimensional maps of those signals. And in here you can see the map which represents the dependence of diffuse reflectance on distance from the source and on wavelength. So we are getting the entire spectral information with one simulation. And you can see in here the simulation results for typical physiological parameters of tissue, such as blood content, water content, melanin contact, and thickness of epidermis layer. And in here you can see that melanin and blood absorption bands are clearly visible. And you can see if we are moving from large blood saturation from low saturation, then two absorption peaks of desoxyhemoglobin are shifted to one peak for desoxyhemoglobin. Okay, sorry. So here are similar results for different blood content of tissue, and we can see that the efficiency of absorption in the blood absorption band increases with the increase of blood content. So in here you can see the dependence of diffuse, the maps which show the dependence of diffuse optical spectroscopy signal on different tissue parameters, such as saturation, melanin content, blood content, and epidermis thickness layer. Actually, this is quite important parameters because fast reconstruction technique doesn't account for epidermis. However, it affects the signal, and in my talk further I will show about the effect of epidermis thickness on reconstruction of optical properties of tissue. And in saturation dependence map, you can also clearly see how two absorption peaks of oxyhemoglobin are transferred into one absorption peak of desoxyhemoglobin. Actually, this slide shows some technical information about our Monte Carlo simulation, which we employed here. You can see that actually the calculations for different spectral regions require different times, and actually the longest time is required to calculate a red band of the measurement spectrum, because for blue range the absorption is quite high, so for green range there is a moderate scattering absorption, and for red light it's quite high absorption and very low scattering, so actually we have to process a lot of scattering events, so it takes quite a long time. And if we analyze the speed of our Monte Carlo simulations, we are using parallelization of the algorithm. We can achieve the speed of 2 millionths of photon per second using AMD hyper-thread processor. And actually, if we are comparing the speed per each thread, that you can see that even old processors give comparable speed with the modern processors. But of course, the number of threads increases for modern processors. Now let me move to the analysis of probing depths in diffuse optical spectroscopy. Using Monte Carlo simulation, we can trace the trajectory of each photon, so we can exactly build a maps of photon trajectories that contribute to some particular detector. And here you can see this kind of maps for different source detector separation, and we can see that the increase in source detector separation increases the probing depth of the technique. And using massive Monte Carlo simulations, we actually build the spectral maps of probing depths for different source detector separation. We can see the general trend to increase of probing depths with increase of source detector separation, and also we can see the trend for increase in probing depths with increase of wavelengths, because when we are shifting to red and infrared ranges, the absorption becomes lower, so the probing depth increases. And for small source detector separation, you can see that the effect of blood is extremely pronounced. So in order to more exactly analyze how can we choose the spectral range for extraction of some particular parameters, we performed an analysis on the accuracy of saturation reconstruction using different spectral ranges. So we analyzed visible range, near-infrared range, and entire range. And actually, we got the results that for different ranges, in different situations, the accuracy differs significantly. So for small source detector ranges, and for small blood content, and for small thickness of epidermis, visible range is preferable, so it provides better accuracy. For high blood concentration and large source detector separations, it's better to employ near-infrared range. And the entire wide spectrum also provides some good results, but not for large blood content and for large thickness of the epidermis. So, and now let me move to the third part of my talk. How can we apply analytical models to improve the quality of reconstruction? The results I have shown you before are based on the classical diffuse approximation of radiative transfer equation. So it employs this kind of green function, which is quite easy to use because it has quite elegant shape. Actually, this equation is obtained for infinite medium. And actually, when we are measuring some biotissue, we have semi-infinite medium, and this should be taken into account. And in 1992, Farrell proposed the analytical solution. It was based on modification of diffusion approximation, which has this kind of shape, and you can see that this is quite long formula, which was still used by different researchers. This formula was obtained for total reflectance, so it accounts all the photons which exit the medium. Actually, we improved this equation, so we got a solution which also allowed to account for limited numerical aperture of the detector, which you can use, because most of the source detector configurations use fiber optics, and you have limited numerical aperture. It's quite important to account for it. So we got this kind of expression, and we used it to compare with two solutions, and we tried to analyze what are the advantages of this model. In this slide, you can see the solutions of dependence of diffuse reflectance on source detector separation. Large screen shows you logarithmic dependence, and the inlay shows the linear dependence of the small source detector separation region. And in here you can see that dots show the results of Monte Carlo simulation, which we consider as ground truth. And you can see that when we are looking at logarithmic scale, both analytical models that account for semi-infinite medium provide quite good results. But if we go to small distance range, then we can see that actually our model, which is shown as a refined solution with red line, it provides much better accuracy as compared to both previous model by Farrell and the model by traditional green function. And in here we show two curves because they are for different ranges. So this is for visible range and near-infrared range, which are characterized by different ratio of scattering and absorption. So in here you can see that our new model provides better agreement as compared to other models that are considered in this study. So in here you can see the results of application of our model for simulation of reflectance spectra for optical properties that are close to real ones, that are based the study by Salomatina. And so you can see that for different blood content and for different source detector separation, we can get a pretty good agreement of model with Monte Carlo simulation, which we consider to be the ground truth. So, however, as I already told, we have to account the presence of epidermis layer, so top layer on the surface of the tissue. And in here you can see the first graph shows you the difference between uniform medium with uniform optical properties and two-layer medium with top layer that has the properties of epidermis, so there is some difference. And we also analyze the ratio of the spectra, which are important for self-calibration approach. And in here you can see that actually the diffuse approximation give quite high discrepancy with the results of Monte Carlo simulations, which are shown with dots. So it's not very good to use this approach because it gives you some error. And both numerical models provide quite good estimation, and actually there is a little difference, and our refined model is closer to the Monte Carlo solution as compared to conventional model. And you can see that another curve, which is also shown by points, shows the Monte Carlo simulations for two-layer media that account for epidermis thickness. And also there is some discrepancy, so actually the presence of the top layer somehow affects the measurements. And in here we try to perform reconstruction using three considered models. So for the uniform media case and for the case of two-layer thickness. And you can see that actually, first of all, two layers for the semi-infinite model allow for separate reconstruction of absorption coefficient and scattering coefficient, and our refined model is more accurate as compared to conventional model. And for with diffuse approximation, we can extract the effective coefficient, and then here you can see the comparison of extraction of effective coefficient using Green's function and other analytical models. And of course, analytical models provide you more accurate results. So the presence of epidermis induces additional error in reconstruction of absorption coefficient and scattering coefficient. However, what is important, if we are having a look at effective coefficient, the both analytical models are almost insensitive to it. So it means that if we are reconstructing effective coefficient, we can forget about epidermis. It doesn't affect it, and I think this is quite good results, which shows that if you are reconstructing effective coefficient, then you can neglect the presence of the epidermis. So now let me move to conclusions. So we derived some simulations using Monte Carlo, and we developed a novel analytical model. We analyzed the different spectrum ranges for reconstruction of saturation, and finally we demonstrated that if we are using complicated refined models that account for semi-infinite medium, then we can perform reconstruction of effective coefficient, and it's insensitive to the presence of superficial layer, which is basically epidermis. So this is our team that contributed to this study. I would like to acknowledge the Center of Excellence, Center of Photonics, and thank you all for your attention, and I will be happy to answer your questions.
Thank you, Mikhail, for a very interesting presentation, and we have time for several questions. Yes, Alexei, Dr. Semyonov, please.
Thank you very much for your presentation. My question is about the, your needs and your demand for the computing equipment. So you provided the diagrams of the power, yes, of the values of the different processors. So what's your what will be the ideal for you, and will be the use of supercomputers that are appearing nowadays pretty often could help you in improving the speed and the quality of your calculations?
So actually, one of the ideas of the slide is to show that actually the problem is fully scalable. So if you compare the efficiency for one thread, it's almost the same for old-style processors and for modern processors. And you see that this modern Threadripper processor with 128 threads, its working frequency is even a little bit smaller as compared to single thread of older processors. This is the idea that you can use several normal computers, or you can use some supercomputer. And the only main parameter is the total number of threads. So it's fully scalable problem.
A small additional question. I heard that sometimes in calculations of molecular dynamics, they use the set of video cards processors, and they say that it helped them a lot, even comparing with modern processors. Have you tried it?
Actually, this is in process, and I hope I will show you the results on the next time. So actually, there is a potential of further improvement using graphical cards, but in graphical cards you have to be quite accurate with shared memory, because in here you can separate the memories, and if you would like to calculate some three-dimensional data, this may make the simulations with graphical cards less efficient if you have to interact with general memory each time.
Thank you. More questions? Okay.
So the question about the refined model.
Okay.
Is it about one layer or a multi-layer system?
Actually, it is for one layer so far.
And can it be transferred to the multi-layer system?
Now we are developing further improved system which accounts for another layer. But as I already shown in this presentation, for some tasks you can use even one layer model, and for reconstruction of efficient coefficients, the presence of the superficial layer doesn't affect that much.
And can this refined model for semi-infinite medium be combined with the infinite medium? So you have the first layer with exact depth, and then you have the semi-infinite medium.
But actually, I didn't got really your idea, because we are considering semi-infinite medium, so there is only one border, and you have air above it, and below it's all tissue. And
another layer upper the semi-infinite. Just a thin layer like film on the cover.
Yeah, and I showed you that for some applications this film doesn't affect the result. Or that's
Okay,
it.
colleagues, you can discuss it later.
Yeah, okay.
During the coffee breaks. Again, another questions from audience? So I have a methodological question to you. You demonstrate the depths of probing that you obtain in Monte Carlo simulation. Could you please? Yes,
like this
one.
Yeah.
I can see the curve that is demonstrates the probing depth. Yes.
Yeah.
How this curve was calculated?
It's median depth. It's median depth.
What does it mean, median?
Mediana.
Okay.
Actually, this map shows you the distribution of probing depths. So at particular source detector separation and particular wavelengths, there is a distribution of contributing photons over maximal depths that it reaches in the medium. And the curve shows the median of this distribution in each point.
Okay, and just a little question about one of your main results about analytical model. As you said, you use diffuse approximation in your model. Did you apply some other limitation?
So actually, we used classical diffuse model, so with this kind of green function, and further assumptions that there is a semi-infinite medium and there is some limited numerical aperture can give you this kind of solutions. So some additional assumptions are applied to diffuse approximation to get these more complicated solutions.
Okay. Thank you very much.
Thank you so much. Thank you
For nice presentation. And It is my pleasure to announce the next speaker, Professor Ma from Tsinghua University, and he will give a talk with the title Pathological Feature Extraction by Muller Microscopy and Machine Learning. This will be online invited presentation. Professor Ma. Hello? You can share your presentation. Yes. Okay. Please share your screen.
马老师，
Okay, and please switch to full mode. Full screen mode. Unfortunately, we can't see your presentation. Could you please switch to full screen mode?
马老师，您在说话吗？好像没有声音。然后您现在可以看到这个您那个麦克风的那个位置有一个像那个小小的箭头往上的，您重新选一下看一下。
Hello, can you hear us?
Yeah, I think he can hear us, but he can. We cannot hear.
So he can. His microphone is
So
doesn't
please
work. Okay.
wait for a minute.
Okay. Okay. We will wait for a few minutes. Ну, can you share your screen again and try to speak something? Unfortunately, we can hear you.
He's trying. He's trying.
Okay. Вот сейчас мы.
Hello.
Hello, hello.
Can you hear me?
Very good.
Can you hear me now?
Yes, we can hear you.
Oh, all right. Okay. Can you, let me share the screen?
Yes, you can have rights. Please share.
All right. Okay, I'll share my screen.
Yeah.
Okay.
Okay.
Do you see my screen?
Yes, we can see your screen, but we can see your presentation.
Okay.
Please show your presentation now.
Okay, right. Shall I start? Can I start now?
No. We can see your presentation on the screen.
Can you hear me?
Yes, we can hear you, but we can see your presentation.
All right. Okay. I start now. All right. Yeah. Thank you very much.
No. Sorry, but we can't see your presentation on the screen. We cannot
You
see.
cannot.
Yeah, we cannot see.
You cannot see my presentation in the screen. All right.
No, okay. We
Uh,
cannot see. Yeah. Please try to share your screen again.
now?
No. We can
How
see
about now?
we can see your screen with the sharing program, but there is no your presentation on the screen. We cannot see your PowerPoint presentation.
You cannot.
Yes, we cannot see.
How about now?
No. Yes, please go to the screen.
Sorry. Okay, I'll come back. Now?
It is some delay. Yes, we can. Yes, please.
Is that full screen?
Unfortunately, we can't see the full screen mode on your for your presentation.
It's not, or it is full screen.
Well, okay, you just switch your slides inside the PowerPoint user interface.
All right. Okay. Yeah.
Okay.
All right. Okay. Yeah. I apologize for the delay, and we tried. I thought everything was okay, but anyway, yeah. So thank you very much for the organizer to arrange this meeting and let me present my recent work. My name is Ma Hui from Tsinghua, Shenzhen, and today I'm going to talk about polarization techniques in pathology. Why I cannot
Professor Ma, just yes, it is good. It works. We
all
can
right.
see
Okay.
another slide.
Okay. That's fine. That's fine. Okay. First, a little bit about polarimetry. You see, as we know, when we describe a polarized light, we need to use Stokes vector, which contain 4 components to describe the polarization states. And when we need to describe the polarization property of a complex media, we use Mueller matrix, which is a 4 by 4 matrix, which encode a lot of information on the optical and microstructural features of the complex sample. So our goal is to measure the Mueller matrix. And for a complex sample, as we are dealing with in biomedical sector, so there are a lot of scatterings in there. We know actually a change of polarization and polarized light interact with the sample. The change of polarization is sensitive to the size, to the refractive index, and to the shape of the scattering particles. So there are a lot of more advantages in polarimetry, so it's easy to adopt, and more importantly, we get a lot of information from the Mueller matrix, specifically super resolution information in the scale below the wavelength unit. In my lab, actually, we developed a series of Mueller microscope, which is a polarization microscope in transmission mode, in reflection mode, in upright configuration, and invert configuration. So we also use those microscopes in hospital, collaborating with medical doctors to find real applications. So once you get the polarization images, the key challenge is how you extract useful information which will be helpful for the medical doctors. So, the easiest thing is you use the matrix components, matrix elements, or you can use parameters, we call it trigonometry basis parameters, which are functions of the matrix element, but with more specific physics properties, physics meanings. Or we can use the combination of parameter basis parameters to get more specific description of the microstructural features. So with those parameters, we can actually, you see, using different parameters, the same image looks different because different polarization features will highlight different microstructural features. So this is the easiest uh, technique. So we use these kind of techniques on different samples, so we can segment different microstructural features and achieve quantitative assessment of a specific pathological feature. Or instead of using a parameter basis parameter, single parameter, we can use the combination of those parameters, and we use deep learning, supervised learning, then characterize the different microstructural features uh, 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 of a specific microstructural feature. So that the earliest work, and we have been using this kind of technique in different applications. The basically we segment the pathologist segment a specific area, and by collecting a large number of labeled areas, we use machine learning to train a model which allow us to find the specific polarization feature to represent to highlight the segmented or the pre-labeled area. So that is what we have done in previously. And also, instead of using the polarization features at a pixel level, we can also use polarization feature in the image level. So by finding different polarization parameters which form different images, we can also combine polarization feature and texture feature in the image, polarization feature pixel, and texture feature. Uh, uh, I found get a pointer. Sorry, and the texture feature in the images combine two to identify specific microstructure. But today, I'm going to talk about something else, which is we use unsupervised learning to find polarization features in a complex Mueller matrix image. So the idea is different from supervised learning. In the supervised learning, you need to supervise, which means you need to identify something which is known, which can be used as a standard. And then you need a large number of samples to train a model and to find similar features from unknown. But in pathology, it is not trivial to get very good quality labeling by the pathologist. So we changed the idea. We see why don't we use clustering? So we use clustering technique try to extract features from the polarization image. So this is our first try, and so we use the simplest clustering technique, which is called k-means, and then we can cluster the pixels in the image, the mirror matrix image, as 4 classes. And then we can project those pixels from those 4 classes as images, you can clearly see the different clusters correspond to different microstructure features in the image. Some of them correspond to the wall, the cell membrane, or some of them correspond to the nucleus of the cell. So then we can take one step further, which is we treat real samples, we try different clustering techniques, and we can identify from the real sample what cluster corresponds to what. So in this some example, we actually did liver cancer sample. So we do polarization image, imaging, and we take clustering of the pixels. You see, they form approximately 6 groups: 1, 2 at the left, 3, 4 in the middle, and 5 and 6 in the right. So, and we can, with the help of the medical doctors, we can identify those 4 clusters correspond to normal and the cancer's nuclear, normal and the cancer's cytoplasm, and the normal and the cancer's fibers. So, and even on the bottom left, for the nuclear, we can further cluster it into different clusters. So by doing that, we can decompose the complex biological sample into components of simpler structures, such as nuclear, cytoplasm, and fibers, such as collagen fibers. So this is, and then we can use the different components, find out how they change during the cancer process. So this is one technique developing. And the second one, actually, we borrow the superpixel concept from the image people. So we can actually, by calculating superpixel polarization space, we can obtain a smaller number of superpixels which approximately present the polarization feature of the sample. So it's like we use, say, 1024 superpixels to represent a complex polarization feature in 16-dimension polarization space. So using those simple pixels, we can realize something we call label spreading. So such as this. So we can take millimetric image and then we segment a small area which medical doctors know something is wrong. And then we calculate the contribution of different superpixels to the labeled area, and we can weight for this specific superpixel. And by continuing this, we have weight for every superpixels. Each superpixel represents a polarization feature. And then by doing that, we can assign weight to every pixel. Then using this pixel and weight, we can reach label spreading, so we can label similar areas with, we can label areas with similar microstructural feature as the pre-labeled area. This is the result. So we can start from a small labeled area and we can extend the labeling, and then we can label the entire area, starting from the small area, small pre-labeled area. And from one sample, we can extend the labeling to another sample and then to another. So in a small time and quickly extend the labeling to a large number of pathological samples. And then we also provide a software which provide a platform to help medical doctors communicate with our student or the technology people. We can work with the medical doctor. We can analyze the pathological sample together to identify pathological features. So that's so that's what we do. Yeah. So now we already have a platform which allow us to use polarization image to identify pathological features quickly from a small number of relabeled samples. So that's what I'm talking to talk about today. Now I'm open to questions. Thank you very much.
Okay, thank you very much for your talk. And we have a few minutes for questions from audience. Does anybody have questions? So I have one question to you about Mueller software. Did you apply this software in clinical practice, or it is just planned?
Well, not really, not yet. It is quite new. We work with the medical doctors. We still need, but maybe it can help the medical doctors to identify something, but it's not really clinical application. It's still research at the moment.
Okay. Thank you very much. Is any
Thank
okay?
you.
Thank you very much. Let's thank speaker. And next speaker, Dr. Olga Cherkasova from Institute of Automation and Electrometry of Siberian branch of Russian Academy of Science, and she will be delivered the talk Raman spectroscopy of body fluids for glioma diagnosis. So, Olga, please.
First of all, I would like to thank the organizing committee for inviting me to this conference. Так, сейчас мы разберемся здесь. Куда? Вот так вот, да? Не двигается. This is the structure of my report. I will speak about glioma and about ordinary medical. А я не могу громче. Кнопка. На микрофоне кнопка? Давайте кнопочку. Все работает. Все нормально?
One. Yes.
Давайте. I will give some characteristics of glioma and ordinary methods for glioma diagnosis. Then I will speak about tissue and liquid biopsy and about biological glioma biomarkers. I will speak about our experiment with glioma model and about samples from people with glioma pathology and about Raman spectroscopy and our machine learning methods. And at the end, I will give some conclusion. The most common brain tumors are gliomas, which begin in the glial supportive tissue. And glioblastoma is one of the most aggressive, fast-moving, and deadliest types of glioma. One reason for the poor outcome of glioblastoma may be delayed diagnosis and treatment, but knowledge about early stage of glioblastoma is limited. Exist different tests and procedure used to diagnose glioma, and first of all is a neurological exam was checking your vision, hearing, balance, coordination, strength, and reflexes. If there is a difficulty with a certain task, it might be a clue that there could be a brain tumor, and the next step is the different imaging method, and magnetic resonance imaging and spectroscopy is one of the methods which often used in medical practice. Then method is computer tomography and positron emission tomography imaging. This method used special radionuclide substances. So, for example, you can see in this used 18 fluorodeoxyglucose, and this is glioblastoma, and this is MRI imaging as compare. And the next step is tissue biopsy, and you can see gematoxylin and azine staining glioblastoma, and you can see unclear bodies, and all these methods cannot be used for diagnosis of glioma at an early stage. Disturbance in brain metabolism during glioma development is reflected in changes in the composition of body fluids. The different substances can be go to blood. The determination of molecular markers in body fluids has great potential for the early diagnosis of glioma. And in this picture, I give a comparison of traditional tissue biopsy and liquid biopsy. Tissue biopsy is invasive, longer time, low sensitivity, not real monitoring of drug response, and so on. But liquid biopsy has many useful properties, and control of glioma molecular markers in body fluids has a potential to become equivalent to studying glioma tissue. It is important that similar molecular markers can appear in body biofluids at the early stage, opening a way to an early diagnosis. Optical spectroscopy often used to oncology diagnosis, and among them infrared and Raman spectroscopy are complementary technique. Raman spectroscopy can be more effective than infrared spectroscopy due to the higher sensitivity to CC bonds, and to the aim of our research application of Raman spectroscopy of body fluids for glioma diagnosis. This is our experiment. So we used the model of orthotopic xenotransplantation of U87 human glioblastoma cells in immunodeficit mouse, and the dynamics of the tumor growth were studied by means of magnetic resonance imaging, and you can see glioblastoma in brain mouse, and we analyze blood plasma through 7, 14, and 21 days after tumor cell inoculation. We analyze the blood plasma of mice by Raman spectroscopy and machine learning methods, and then the proven approaches were used to analyze the blood plasma of patient with glioma. We analyze patient with glioma different grade and patient with skull craniotomy defects, and donors samples was obtained from Novosibirsk Research Institute of Traumatology and Orthopedics. We use such Raman spectroscopy and special plate for samples. Each sample was dropped with a volume of 10 microliters. Special aluminum plate and measurement were carried out after complete drying of samples. This is experimental Raman spectrum. And we use in this picture you can see implemented machine learning pipeline for Raman spectra analysis. Raman spectra were normalized by maximal value to be able to compare spectral intensities. Then we applied different methods to remove background. Principal component analysis was used to extract informative features and support vector machine, random forest, and XBoost were used to build prediction data models. And you can see average Raman spectrum. Мышку не видно, да? Да, я поняла, да, это мышка вертела. Average Raman spectra of each experimental groups, and this average spectra of experimental and control groups for every experimental group for 1 week after Thomas-cell inoculation, 2 weeks, 3 weeks or 21 days, and 28 days after Thomas-cell inoculation. And you can see the difference in average spectra is insignificant, and so we used machine learning method. You can see data for third group after Thomas-cell inoculation, 21 days. You can see explained variance of principal component for third group. The first 4 components, which make up 98% of variance, are the most informative and significant for group. And in this picture, you can see projection of the data on various main component. Main component 1 and main component PC1, PC2, PC1, PC3, PC2, PC3, and PC2 and PC4. And you can see that separability of group is based visually for the second principal component. So most informative frequencies corresponding to the second principal component. And you can see loading matrix analysis and selection of informative frequencies. Analysis of loading matrix for the experimental third group and control groups. Since the class separability is carried out according to the second principal component, therefore the following ranges will be informative. This frequency, this and this frequency for this principal component too. You can see predictive model and its verification by the method of characteristics curve. Mean rock. Mean rock curve for linear support vector machine. and you can see excellent separation group. AUC is 0.94, and linear separability between third group and healthy mice was determined by the linear support vector machine with 86% sensitivity, 83% specificity, and 84% security. And in this slide, you can see the informative frequencies for this classifier, and this is frequency. More important for differentiation group. Changes in Raman spectra depended on the volume of glioma, and you can see that volume changes are linear during our experiment, and so we use a non-linear regression model, and the quality of model was estimated by mean and standard deviation of absolute area and determination coefficient, and you can see these parameters, and then we analyze more informative Raman spectra features associated with tumor growth, and you see these features, and with the progression of tumor, an increase in the contribution of lactate, tryptophan, fatty acid, and lipids are observed. In our study, tumor volume vary from 0.002 to 0.098 centimeter in cube at different stage of the experiment. So that is several order of magnitude lower than in paper number 2. And we can conclude that Raman spectroscopy of blood serum can be used for early glioma diagnosis. More information can be found in our recently paper. Then we analyze patient blood plasma using Raman spectroscopy and machine learning. We analyze two groups. Glioma patient versus healthy donors, case 1, and glioma patient versus skull craniectomy defect patients. So this is average Raman spectra for each groups, and we use the same machine learning pipeline as we found for experiment with mouse, and you see predictive model. For first case, you have excellent separation group patient with glioma from healthy donors, and in second case, we have satisfactory separation group, and we also obtain informative frequencies. You can see frequency number 1 and number 2. The greatest contribution of the separation group is made by informative frequencies in the range which correspond to stretching CCC and bonds in molecules of proteins, carotenoids, amino acid, tryptophan, tyrosine, and phenylalanine. And this is average spectra of cerebrospinal fluid, red line, and blood plasma of patient with glioma. This only the first result, and we can see that some spectral features have different intensities, and this difference depends on composition of analyzed liquids, and this work are continued now. And in conclusion, Raman spectroscopy of blood serum can be used for early glioma diagnosis. Raman shift correspond to molecule of proteins, carotenoids, and some amino acids, and Raman spectroscopy of human blood plasma and cerebrospinal fluids can be used for the diagnosis of glioma and allow to monitor changes in the state of brain tissue during to its development. And this is our acknowledgement, and thank you for your attention.
Thank you very much for the presentation, and several questions from audience. Professor Priezhev, please.
Thank you very much, Olga Pavlovna. My question is, so you showed that there are markers of glioma in bodily fluids, and Raman spectroscopy is sensitive to these markers. So question is, how specific are these markers? Is it possible that they are markers also for other types of tumors in the body?
In our research, we compared blood plasma patient with glioma and patient with different pathologies, and we obtained that we can differentiate this group with high specificity.
Other pathologies, non-cancer or cancer?
Yes, non-cancer. This non-cancer, but every pathologist has special biomarkers, and I think that machine learning method can analyze to differentiate a group of different pathologies and different grade of glioma.
So this is specific, right?
This is specific, yes.
Okay, next question. Okay. Dr. Bartashov.
Have you some specific information to what molecules, what molecular group are different in the spectra to, and what changes in molecular structure during the disease development?
Raman spectroscopy. We have experiment with our animals, and we see different informative frequencies for every stage of experiment. In our work, we not analyze specific molecular structure, but we see spectral picture from on every stage of experiment. We have special spectral features. Yes.
Okay. Other questions? Just one question about what was the volume of the data that you used to teach your machine learning algorithm? How many patients or samples did you use for that?
So I have special slide for this work. В смысле, for this question. Of course, for creation special diagnostic method for medicine, we have more samples, but we used such approach. We analyze many spectrum from one patient, and so we have a big massive of data.
How many patients?
You can see in this page.
24 patients. Okay.
24 for different grade, 23 for this pathology, 21 donors, and we have about 80 mouse.
Okay. Thank you. Let us thank Dr. Cherkasova for the presentation. And we are going to the next speaker, Dr. Marina Shermanova from Volga Research Medical University from Nizhny Novgorod. And she will deliver the talk, Microviscosity of a Cell Membrane and its Implication for Cancer Treatment.
Hello everyone, it's a great pleasure for me to give a talk here, and first of all I would like to thank organizers of this meeting, Professor Priezhev and Professor Tuchin, for the invitation to speak here. And today I'm going to tell you a little about the viscosity of plasma membranes and its relation to cancer and chemotherapy. Here's the outline of my presentation. I will give a short introduction into the topic of biophysics of membranes, and then I will describe materials and methods we used, and then I'll present you some results on chemotherapy of cancer with different drugs, all the drugs used in the clinics, and then I'll conclude with a summary. Viscosity is a property of liquids. It's a reciprocal of fluidity. It's quite the same. And just to feel what viscosity is, just imagine how to flow water and honey. And biological membranes is a kind of liquids, and their viscosity lies somewhere between water and honey. It's like an oil. You know that biological membranes are the complex structures, and the term microviscosity is used is applied to them to underlie that the measurements are made on the microscopic scale, this microviscosity. It's a complex structure. It's a lipid bilayer with a lot of embedded proteins that include different enzymes, transporters, receptors. And the main function of the cell plasma membrane is to separate the internal content of the cell from external environment, so to preserve cell integrity and homeostasis. And another function is to support support the activity of embedded membrane proteins. So the physical state of the membrane is very important for cell survival and homeostasis and integrity and so on. And microviscosity of cell membranes depends on different factors, including temperature, hydrostatic pressure, and so on. But the major factor that determines viscosity is its lipids composition of the membrane. And there are some lipids like cholesterol, saturated fatty acids in the tails of phospholipids, and sphingomyelin that increase viscosity of the membrane. And other lipids like unsaturated fatty acids in the tails, phosphatidylcholine, decrease it. Very little actually known about microviscosity of cellular membranes, and especially in cancer, and even less is known about its relation to chemosensitivity and to chemotherapy. Also, it is intuitively understandable that viscosity can have a relation to chemotherapy because it's a barrier, it's the first barrier in the cells that prevent the penetration of the drug into the cell, right? So in support of this, some studies actually demonstrate that the more viscous membranes are observed in more resistant, chemo-resistant cancer cells. Yep. And another link between viscosity and chemotherapy is that fast death receptors that initiate apoptosis of cancer cells are located in the plasma membrane. So fluidization of membranes activate apoptosis. And another very interesting and emerging field in anti-cancer therapy is so-called the lipid therapy that is aimed to manipulations on lipids in cancer cells to improve the efficacy of chemotherapy. So the purpose of our studies was to analyze the changes in microviscosity during chemotherapy with different drugs in different cancer models, including cell monolayers, multicellular spheroids, and animal tumor models, and to correlate these changes with lipid profile of cancer cells and responsiveness to the treatment. And the specific questions we asked here were, does in general chemotherapy induce any changes to cell membranes, and how common are these effects for different drugs, and what does it mean for cells, and what are the reasons for these changes? To measure viscosity, we used so-called molecular fluorescent rotors. These are small organic molecules with a key, the key feature of these molecules is a capability of rotating. So the higher viscosity in the microenvironment is, the more quickly, the more fast the rotation proceeds. So the fluorescence lifetime of this molecule increases with increase of viscosity. And we can visualize fluorescence lifetime of these molecules using fluorescence lifetime imaging microscopy. So using these probes, we can have a map of viscosity of plasma membranes because this probe is located there. So we can do this on living cells, on living tissues, and use one calibration for different systems. This is very important to quantify this value. To obtain fluorescence lifetime images, we use two different systems, LSM laser scanning microscope 880 from Carl Zeiss and multiphoton tomograph MPT flex from Yenlab company. And both systems are equipped with 2-photon laser for excitation and a FLIM TCSPC time-correlated single-photon counting model for obtaining FLIM images from Becker-Higel company. Here is a design of our study. We used different cancer models, cell monolayers, 3D tumor spheroids, animal tumors, and we treated cells with different chemotherapeutic agents, including cisplatin or oxaliplatin, 5-fluorouracil, paclitaxel, and then in vitro studies we confirmed, we verified the lipid composition of membranes using chemical analytical techniques, the TOMF-SIMS. And what I should mention here that all the drugs we used are in clinical protocols for cancer treatments, but none of these drugs do not involve plasma membrane in their mechanisms of action. So the target of this drug, either DNA as for cisplatin, oxaliplatin, or 5-fluorouracil, or microtubule as for paclitaxel. So the effects I'm going to show you later are likely associated with any indirect effects of these drugs on the cell membrane. Previously, we developed the protocols to image and quantify microviscosity in different models. We did it with monolayered cancer cells, with spheroids in vivo, and interesting things we concluded from all these previous studies that viscosity of cell membranes is quite stable and very specific value for each cell types. So different types of cancer cells have different microviscosities. It is in the range from roughly 300 to 500 centipoise, but it's quite specific. It doesn't depend on proliferation activity, on metabolic activity of cells, or even on the state in which, on the conditions in which the cells grow. For example, we see quite similar values of microviscosity in cell grown as monolayer and in the tumors in vivo obtained from these types of the cells. This was a quite surprising results for us. And then in one of our first experiments on chemotherapy and viscosity, we treated cancer cells with cisplatin and obtained FLIM images in dynamics. And what we found here is that viscosity decreased at early time points of the treatment, but statistically insignificant. And then at later time point, it statistically increased. And this increased value of viscosity, elevated viscosity, preserved even if we removed the drug from the culture media. Then we were wondering to know how specific are these changes for different cell types, and used different cell lines. That was CT26, this was HeLa, and we used different drug concentrations to analyze, let's say, dose-dependent effects. And what we see here is that in different types of cancer cells. We see also decrease of viscosity, then increase. There is no dependence on the drug dose. Then we tested the same therapeutic regimens and did these experiments on multicellular structures, 3D tumor spheroids. You know that spheroids are multicellular in vitro model of tumors. It's a kind of model intermediate between cellular monolayer and in vivo tumors. It's very useful model to visualize cellular heterogeneity because the outer layers of cells in the spheroids proliferate faster, and the cells inside the spheroid are, let's say, quiescent. And we didn't see, first of all, we didn't see any differences between viscosity in the cells outside of the spheroids and inside. It's the same value. But during the treatment, we observed the same effects of cisplatin on cancer cells. Again, we see an increase of viscosity at the later time point of incubation. To ensure that the effects that we observe on viscosity are associated with the response of cancer cells to the treatment, but are not associated with a general stress, let's say, or associated with chemotherapy. So we performed the experiments using chemosensitive, chemo-resistant cells. Yeah. This was done in vivo. So we had resistant cell lines and generated tumors from these lines, which were also resistant. And we found that indeed the effects on viscosity that we see previously, previously associated with response, because only responsive tumors have an increase of viscosity, but resistant tumors didn't change the viscosity of plasma membranes. Here just a verification of that the therapeutic effects to take place in this study. Next we tried to analyze the reasons for the changes of viscosity, and the first possible reason was just direct interaction of the drug with the plasma membrane. To look at this, our collaborators from Imperial College London, this is a group of chemists, Dr. Marina Kuimova, they did the experiments on lipid vesicles obtained from different lipids, which mimic the composition of plasma membrane. And as expected, the addition of cholesterol and sphingomyelin to this vesicle increased plasma membrane, increased viscosity, but the addition of the drug to this vesicle didn't change it. So that means that it is not a direct interaction of the drug with lipids of the membrane. Okay. So the next question was, what this can be a consequence of the changes in lipid composition. To analyze this, we use the method of the time of flight secondary ion mass spectroscopy. This part of the study was done in collaboration with Dr. Alexander Gulin from Institute of Chemical Physics, Moscow. This is a surface-sensitive analytical techniques which analyzes composition of the membrane on the surface. So the typical mass spectra for different components are shown here. Among the different lipid components of the membrane analyzed with these techniques, we precisely looked at those that can have any effect on plasma membrane viscosity. And what we found here is that indeed, among different changes in lipid composition, there are some that correlate with the changes of plasma membrane viscosity. We see here the increase in phosphatidylcholine, and increase in cholesterol, and decrease in monounsaturated fatty acids. And so these changes the changes in plasma membrane viscosity can be associated, at least partially, with these changes in the lipid composition. So how specific are the changes for different drugs? Yeah? We used here 5-fluorouracil, this is a drug with different mechanisms of action. And what we found here, also we see the changes in sensitive cells and didn't see any changes in resistant cells, but the changes were quite different. All we see with 5-fluorouracil were temporal changes. They were observed only during first hour after addition of the drug. Shown here, first we see again a decrease in viscosity and then an increase, and then the viscosity returned to the initial level. And again, these changes were also associated with changes in the lipid compositions, but they were quite different. We see lower level of unsaturated fatty acid and decreased phosphatidylcholine, which partially again explained the increase of viscosity at 1 hour incubation time point. The last drug that we tested here in this study was paclitaxel. Paclitaxel have completely different mechanism of action. It's antimicrotubule agent. And what we see here that the effect on it also have an effect of plasma membrane viscosity, but the effect is completely different. We didn't see any change during 6 hours, and then we see not increase but decrease of viscosity at 24 hours in sensitive cells and no changes in resistant cells. And this also most likely was associated with changes in the lipid composition. At least we see a higher level of unsaturated fatty acid, which could be a possible reason for decrease of cell membrane viscosity. So here is a summary of all the effects of different drugs. What we conclude from these studies is that different chemotherapeutic agents have an effect on plasma membrane viscosity. This is the first conclusion from this study. The second conclusion is that different drugs have different effects on plasma membrane viscosity in terms of dynamics and the type of these changes. And among different alterations in lipid composition associated with the action of chemotherapy, there are always some changes that explain, that correlate with viscosity changes. So, to conclude my talk, I would like to say that we developed a methodology to measure, to visualize viscosity in different models in living cancer cells, in monolayer, and in vivo, in vitro. And chemotherapy affects microviscosity of plasma membrane, and this is associated with modification of lipids, and alterations to plasma membrane accompany the response of cancer cells to chemotherapy. And what is important point here is that these changes in microviscosity should be taken into account during planning of chemotherapy, because different drugs normally used in a combination. So if you have an effect of increased viscosity with one drug, so this may have an effect on penetration of the next drugs used in the combination. So destabilization of plasma membrane can be also considered as a solution to reverse chemoresistance, because chemoresistant cells have more stable plasma membrane viscosity state in terms of viscosity. I would like to thank our collaborators from Baker Hickle Company, from Imperial College London, in Institute of Chemical Physics, and I would like to thank our team, especially Dr. Lyubov Shimolina, who did a lot in these studies, and thank you very much for your attention. I'll be happy to answer your questions.
Thank you, Maria. Professor Priyazhev, please.
Thank you very much for a very nice talk. My question is about the notion of microviscosity. It is very, very complicated notion. This is why people often say apparent viscosity. It very much depends how you average over the volume where you measure and so on. To my notion, actually, biological membrane has different viscosities at different layers, at different depths. So how did you measure? So is it an average over the whole thickness of the membrane, or on the surface, or below, and so on?
Yeah, I understand your question. It's a very interesting point. The depth and the resolution in which you measure the viscosity depends on the instrument you use, right? So the probe that we used is located inside the bulk part of the membrane, somewhere inside, somewhere between the lipid bilayer. So we suppose that we measure at least to the whole depth, because just the depth of the whole membrane is just 2 nanometers between lipids, right? So it's very small value. So we believe that we measure this to the whole depth, but we are pretty sure that we measure viscosity in the bulk of the membrane, because it's even more complex structure that can be imagined, because there are the structure called lipid rafts, which are the complexes of lipids and proteins. So our probe doesn't go there. This is known for sure. So this is a bulk membrane.
Thank you. Professor Kistinyov.
Marina, thank you for very interesting talk. Seems to be microviscosity reflects a number of processes which appear in cancer cell during chemotherapy. And some are specific, some not specific, and from my point of view, that variation of lipid content is not the main action of chemotherapy. And it will be very interesting to find direct relation between specific parameters associated with cancer cell condition, associated with cancer and microviscosity. What do you think about possibility of this task solution?
Yes, this is very for today very complex and very interesting question. We are thinking a lot about this, and for us we did the lipid profile, the lipidome of the cell is very flexible, as we know. Yeah, and the cells react on changes in conditions of culture and different factors by very quick changes in the lipids. They can be adjustable to anything, yeah? But viscosity can be preserved at this. We see this with our resistant cells. So actually the lipid composition between resistant cells and non-resistant cells can be the same, but the lipid composition will be completely different. So it's a very complicated story, I would say. So no one lipid that control microviscosity. It's a kind of interactions between different ones.
My idea is that chemotherapy mostly influence on other components of cell.
Yeah.
And maybe there are relation of such parameters and microviscosity.
If it would be so, we would see quite similar changes in viscosity if we used the drug with the same mechanism of action, or at least with the same target. Yeah, as we observed with 5-fluorouracil and cisplatin, they have the same target. This is DNA, but the mechanisms are different, and different changes in the membrane viscosity we see. So no answer. Thank you.
Okay, one more question, Dr. Semyonov.
Marina, thank you for a brilliant presentation. Summarizing your results on cells and animal models, summarizing results on model objects like vehicles and so on, what do you think, can you say that membrane microviscosity can be a parameter of efficiency of the drug delivery to the cell? Can we say that it's, we can use the parameter to study the efficiency of the drug delivery?
I think the answer is yes, because all these changes in viscosity are associated with responsiveness of the cells. If you take resistant cells, we do not see any changes in viscosity during the treatment. So at least this parameter can be used to differentiate between responsive and non-responsive cells, I would say. Yeah.
Okay, let us thank Marina again.
Thank you.
Thank you very much, and we are closing our session, and now we will have a lunch. After that, we will meet at this room, and we'll continue the Russian-Chinese seminar.
I'm here. I'm here. Hello. Okay, please wait.
Hello, Mr. Chair. I'm Wei Zheng from China, and I'd like to test my PPT. So
Hi. It shows code 1136.
那个小张，你让郑伟老师先试一下，你把你的截图截给我看一下。
我把我的截图发给您是吧？
对，你是什么问题？
你把那个，就你把这块，不是这个截图，是这个。
你分享不了吗？Hello.
Yeah. I want to test my PPT. Please give me the authority. I think she has some problem, and she said he can't share the screen now. Yeah, he's swing Lee. Yeah, that's right. Okay, I test my. I'm Wei Zhen. I just test my PPT. Okay, or Swen Lee.
Hi. We are here to have some problem. We don't have the permission to capture the screen.
Hello, I think Suen Lee has some problem with her computer. Can we switch to Professor Wei Zhen as the first to talk?
Yes. The screen showed code 1136.
1136.
对。Okay, thank you. No, it still showed. Okay, well, I will switch another computer.
Hello, Mr. Chair. Hello, Mr. Chair. Yeah, I'm Wei Zhen, and I think I'm the last speaker. Can we? Yeah. Okay, okay. The last speaker, Mr. Wang Fu, tell me that he is sick. So he says sorry to all the audience. He can't make the presentation. So I think I'm the last speaker. And because Miss Si Wei Li, Mr. Si Wei Li is some time to prepare his computer. I think I can take the time to make my presentation. I mean, I changed the sequence. Can we change the position? I mean, I make the presentation now. Okay. Okay, give me the permission to share the screen. Okay. Hey, I see it. Can you see my? Okay. Okay. So let's start.
Perfect.
Okay. Thank you, Mr. Chair.
Let's start.
Okay, thank you, Mr. Chair, and thanks the organizer give me this opportunity to share our present work. I'm Zheng Wei from Shenzhen Institute of Advanced Technology, Chinese Academy of Science. And my talk is about two-photon microscope of intrinsic signals. So at the beginning, I want to make a brief introduction about the two-photon excitation. Here shows the Jablonski energy diagram. As you can see, this is the two-photon excitation. Compared with the single photon excitation, there is only different here. The photon should absorb two excitation photons simultaneously to jump to the excited state. So due to this, okay, and you will find, okay, only the focal point, only the focal point could be excited and to emit the burst signals. So now we can scan the focal point at X, Y, Z spatial directions and perform the 3D image. So there are many advantages about two-photon excitation, such as the inherent 3D image capability, the superior optical penetration, and the reduced photo damage and high signal to noise ratio, and so on. There are many studies about two-photon microscopes. The other issue in my talk is about the intrinsic optical signals. Actually, there are many types of intrinsic optical signals, and in this talk, I will focus on these two types. The first one is endogenous photons, such as NADH and FAD. They are the metabolic enzymes. The keratin, collagen, and elastin, they are fixed proteins. And the tryptophan, phenylalanine, and the tyrosine, they are the essential amino acid. And hemoglobin also are endogenous proteins, so it will excite with two photon excitation, and it can emit photofluorescence. The second type of intrinsic optic signal is this one, second harmonic generation. It was generated from concentric symmetry molecules. In biology system, the collagen is the major source of SHG. And to make full use of this endogenous, make full use of these intrinsic opticals, we should build some very special two-photon microscopes. Here shows the diagram of our system. This is a time and spatial resolve photon microscope. Compared with conventional system, there are changes at this. We use a spectrograph to detect the spectrum, and we use a TCS-PC, which is used for time-corrected photon counting to detect phone's lifetime. So our system, we can not only capture the X, Y, Z three-dimensional space information, we also can get the first structure and the first lifetime at each pixel. So this is five-dimensional information. With this system, we carry out a lot of biological studies. Here I show one example. This is a hamster. It was anesthetized and the cheek punch was imposed. With our system, we can take the depth result image and have the result. At the beginning, there are some cells, this is epithelial cell, and at certain depths, cell disappear, and you can find many collagen fibers. In some space, we have found blood vessels and some muscle fibers. Emphasize 2 points in this study. The first thing is all the experiment is non-invasive. We didn't do any treatment except the anesthesia. This is the first thing. The second thing, the signal is from the intrinsic signal. We didn't use any extrinsic agent. Maybe some guys want to ask me that how can you identify this blood vessel? I think this topic should attribute to the lifetime detection capability of our system. Because the first lifetime of hemoglobin is extremely shorter than other bronchos. So we can different or we can identify the blood vessels in first lifetime, use first lifetime information. And due to the spectral detection capability of our system, we can identify the muscle fiber and the protein fiber in the spectrum domain. So this is a very powerful system. And to further uh, test its facility, we developed an animal model of epithelial cancer. Is a hamster, and we treated the cheek punch as tissue drug with MBA for about 20 weeks. The tissue will go through stages of hyperplasia, dysplasia, and final carcinoma. And now we take the image from the tissue in vivo, the non-invasive. Here shows the representative result from normal, from low-grade precancer, from middle-grade precancer, and high-grade precancer. The number in each corner is means the depth below the surface. Obviously, you can find the nuclear in abnormal tissue is significantly larger than in the normal tissue. And you will find the system of epithelial cell sick in abnormal tissue. We also can do many advantages because this is a digital image. Okay. We also use this system to capture other clinical samples. This one is from the hamster, and we use this system to image clinical samples. Here is the gastric tissue on clinical surgery, and there are 4 types. The first one is normal, and the second one is CGE, which means chronic gastric with erosion, and the third one is ITA, which is intestinal type adenocarcinoma. And the fourth is CPIM, which means chronic gastric with intestinal disease. And used for lifetime, the spectrum and the morphology features, we can identify these 4 groups very clearly. Another example is also from the clinical surgery. Is the esophagus. And again, with our system, we can identify the normal esophagus and moderate differentiated carcinoma and differentiate statistical. These two cases are published in BOE. Okay, as I mentioned at the beginning, there are many types of endogenous drivers, but currently many studies are focused on the NADH, FAP, and TP. Actually, there are few studies focused on tryptophan and hemoglobin. Why? Because the light source in conventional two-photon microscope system is this one, with a Tesla laser. And this laser can provide the light in wavelengths above 700 nanometers. So this light source cannot excite the difference of tryptophan. To address this problem, we developed a visible femtosecond light source, which is the diagram. This is the diagram of system shown here, and this is the laser. And here shows the characteristics of the laser. You will find the center wavelength is about 523 millimeter. The repeat rate is about 60 megahertz, and the pulse duration is about 150 seconds. One important thing is that the peak energy theta is less than 4%. So this is excellent, good light laser source for two-photon microscope. So we use this laser as the light source to set up this system. This is a short wavelength excitation two-photon microscope. Again, we use the time and spectral result detection as the here, and we can use this system to capture many other endogenous proteins. For example, this is hemoglobin, and you can use this system to efficiently excite the two-fold inferron signal of hemoglobin. The conjugate dependence of the intensity and the excitation level demonstrates that this is a two-photon excitation process. And the first lifetime of hemoglobin is extremely short, which is consistent with previous study. And now we can use the forms of hemoglobin to capture the first image of blood cells. This is first image. And we also can use this signal to capture the three-dimensional network of blood vessels. Blood vessel network as shown here. Again, we also develop a animal tumor model. You can see here, this is the tumor and this is the normal tissue. And we use our system to capture the three-dimensional vessel network. Here shows the normal blood vessel. This is the tumor vessel. Obviously, you will find that the normal blood vessel is very smooth, but the tumor blood is total and leaky. Okay, we also use this system to take images from skin, the year, as skin of the year, and you will find this is the image text from tips from the surface. At the beginning, there are the accidents here, and you will find that this is an heric, and some here also can be clearly identified, and this is blood vessels. And okay, this is text from the in vivo from non-invasively. Here, I want to emphasize that this image is based on the autofront of tryptophan, not from the NADH. It is different to that image actually on before. Okay, because we can take the bronze image based on the tryptophan, we can, again can take different field, different depth image from a specific tissues. We know the esophageal tissue is layered structure. At the surface is capillary layer, and the second is femoral epithelium, and so on. And again, we can take different image from different layers, and they are different, obviously. And we also find the front spectrum and front lifetime at different layers, also different. To magnify, to zoom in some spatial region, for example, the region of tree 3, site 3, and the site 6. From the pattern, we can we think there are the elastic fiber To verify this, we got the first spectrum, just like this, and we also made a spectrum of pure plastic. We find, okay, there is a peak. This is consistent with the consistent with the elastin, and this peak is from the tryptophan. Also, in the region this is the keratin-like area. At the beginning, we think there may be many keratin, but from our data, we found the keratin, the lifetime of pure keratin is very short. It is different to the first lifetime period at this site. However, the first lifetime, this site is similar to the tryptophan. Also, the spectrum is similar to the tryptophan. So we get to the conclusion that the major form in this region is the tryptophan, not the keratin. And in some region, for example, the region 2 or 5, we found the dominant form of the tryptophan, actually. And in site 4, we will find this broad, and we found the first lifetime of this blood vessel is similar to that of hemoglobin, demonstrate that these are the blood vessels. Okay, if we follow the focal point at certain depths, and we can take the time-lapse image of living animals. Here is the video show the movement of blood cells. You can see some blood cells in the vessels. And we also can see the movement of immunocytes. In this case, these are needle to punch skin, and this will cause some immune response, and the immunocyte will move to the damaged site. You can see the movement of the immunocyte here. These results are published on Biomedical Photonics this year.
Dear Wei,
Okay.
you are almost. Oh, okay.
Due to time limitation, I give the short summary. Okay. I think the intrinsic profile can provide wealth information of the biological process, and they have the advantages of unheard environment to investigation, especially in complex systems such as large cell tissues. The second, if we want to exploit the potential utility of intrinsic profiles, a powerful and versatile imaging tool such as spectral result detection, time result detection is the best. Because the intrinsic profile you have the overlap spectrum and unspecific localization. So thanks my group members, thanks my cooperator and founders. Thank you for your attention. You're welcome to visit our site. Thank you.
Thank you very much for very interesting presentation. Yeah, let's thank the speaker. And anyone have some questions? If no,
Thank you very much. I'm Sasha Priyazhev. My question refers to the measurement of blood flow and motion of red blood cells. Is it possible to get some information about the interaction of red blood cells either with the endothelium in the blood vessels or between different blood cells themselves in the flow?
You mean the blood vessel? You mean the different blood cells?
Say again, please.
Sorry, pardon?
No, I was asking about interaction of red blood cells with the endothelium, the cell's layer on the walls, inner surface of the vessels. Can you provide with this technique an information about the interaction of these cells?
The interaction between different cells, you mean?
Yeah.
Yeah. I think this is the case.
Okay, thank you.
Sorry, I think we can image it. We can take the image of cell interaction because we can capture the movement of blood cells and also the blood cells.
Thank you very much, and let's once again thank the speaker. And our next speaker is Irina Dolganova from Osipyan Institute of Solid State Physics of Russian Academy of Sciences. With talk diffuse reflection-based sapphire instruments for tissue characterization during ablation and resection.
So, to work, yes. So, dear colleagues, today I would like to tell you about sapphire instruments for tissue characterization, which are based on the diffuse reflection analysis. So that is an outline of my talk, and I will tell about in particular 3 types of such instruments. The first is the cryoprobe for cryosurgery for monitoring of tissue state. The second is the probe for monitoring of circulatory disorder. And finally, I will tell about diagnostic scalpel, which can operate in different modalities, including the detection of diffuse reflectance. So here is our research group, which is mainly located at the Institute of Solid State Physics. We also work with our friends from Prokhorov General Physics Institute and cooperate with our colleagues from various physical and medical institutes. So this helps us to realize a lot of ideas and concepts in our work. Looking at the properties of sapphire, among high biocompatibility, high thermal and chemical inertness, we can notice a high hardness of such material. Despite the obvious advantage of high hardness, it is also a severe drawback because for mechanical treatment, such hardness make a lot of difficulties. For example, we cannot manufacture the crystals of sapphire with complex shape, for example, with internal channels with diameter less than 1 millimeter, using only conventional mechanical shaping. Therefore, we should use another techniques, and the one possible solution on this problem is to use the crystal growth technique based on Stepanov concept. And here you can see in the laboratory equipment, which is used for manufacturing of such crystals growing from melt. And here you can see 2 schemes of crystal growth, namely age-defined film fat growth technique, which allows us to make profiled crystals with constant profile along the axis, and non-capillary shaping technique, which allows us to fabricate crystals with a variable shape along the axis. So I would not tell about particular the parts of these techniques, but I should mention about the automated crystal weight control, which helps us to minimize the surface and volume defects of such crystals and obtain very high quality of crystals, optical quality, like you can see in this picture. I have mentioned the biocompatibility of sapphire, which makes it a very interesting material for medical applications. But I also want to pay your attention on the high transparency in visible and near infrared range. And high transparency provides us with light delivery to the object of diagnosis or exposure just through the crystal itself. So, assuming the possibility to make crystals with internal channels, we can obtain compact instruments which enable tissue illumination with light and operate for and implement different purposes like resection and dissection, ablation, and so on. also we can do many internal channels, and this immediately apply the specially resolved diffuse reflection analysis. So let's proceed to the particular examples of SAPPHIRE instruments, and the first example is the probe for cryosurgery. And let me remind you that the cryosurgery is the method of pathological tissue destroying using cold. So we apply the cold instrument to the tissue and destroy and induce cryonecrosis inside the ice ball formed in the tissue. Here you can see the very simple submerged type of cryoprobe. Here is the probe itself. It is a simple rod, and one part of this rod is in direct contact with liquid nitrogen. SAPPHIRE has very high thermal conductivity at cryogenic temperatures, and we have shown previously that in contrast to common metals used for cryoprobes, SAPPHIRE enables lower temperature on the surface and inside the tissue, which allows us to obtain bigger volume of ice ball and more effectively induce cryonecrosis. But assuming all advantages of cryosurgery, we should mention about one essential drawback. This problem is associated with the difficulty of monitoring and control the freezing volume and more exactly the freezing depth of ice ball. So it is very important to prevent the freezing of adjacent healthy tissues, and also it is important to obtain cryonecrosis in the whole ice ball freezed volume. So one of the possible solutions of this problem is the application of optical monitoring, which, due to high transparency of SAPPHIRE, can be used in the single instrument. So how it works? Here is a scheme of such a probe. It is a SAPPHIRE rod, but with internal channels where we placed optical fibers for illumination and detection of light. Here is one the longitudinal channel where several fibers are fixed by the metal shield, and these fibers are connected to the light source. And also we have a central placed and connected to the detector. In our scheme, we applied the commercially available system Oxyplex for illumination and detection, and it works using 692 nanometer wavelengths and is frequency modulated. So the frequency modulation allows us to calculate absorption and scattering coefficients of the tissue and estimate the scattering and absorption properties. So when we place our applicator in the contact with tissue, we can consider two states. The first state is the unfrozen tissue, and in this moment we can calculate directly the absorption and scattering coefficients of the unfrozen tissue. And then the second case is the dynamically changed multi-layered system, where here is the ice ball with the parameters of frozen tissue, and here is the adjacent healthy tissue with unfrozen parameters. So we obtain the signals like that from four channels, from four excitation channels of fibers, and comparing the theoretical calculations using these parameters with the exactly detected and measured signals, we can reconstruct the freezing depth of our ice ball. So here is the freezing depth, which increases during the application. So during the experimental approbation, we used tissue phantom contains of gelatin and intralipid. And we can see that our reconstructed freezing depth is in a good agreement with the direct measurement of a freezing depth in such a phantom. But you know that using frequency modulation connects with more complex equipment, more complex calculations, and therefore we have tried to analyze whether we can use stable state mode without frequency modulation to solve the same problem of reconstruction of freezing depth. The probe was shown here, and it has only three channels, one for the source channel and two for detector. We also used the tissue phantom content from gelatin and enterolipid and the well-known expressions for estimation of extinction coefficient, attenuation coefficient, using this expression. Without frequency modulation, we should use another instruments for estimation of initial conditions of unfrozen tissue, but solving this little problem, we also can reconstruct the freezing depth, which also in a good agreement with a directly measured freezing depth. So summarizing this part, I can say that both frequency modulation and stable state mode allows us to reconstruct the freezing depth in tissue up to 9 millimeters, which is quite good. So the second instrument is about cryoprobe, the final part. The final part is connected with the possibility to analyze the tissue state, the presence of necrosis and edema in tissue during the application. You see that we have used a rat, and we induce the cryonecrosis in its liver, and we detect the signal and analyze the scattering coefficient during the application. And you can see that at the initial stage we obtain the increase of the scattering coefficient and then rapid decrease. But for flowering of tissue, we see that the scattering coefficient almost increases. Therefore, we can use such a probe not only for detect the freezing depth, but also for estimate the condition of tissue, whether we obtain the whole necrosis or not. Yes, now we proceed to the second part, that's a probe for monitoring of circulatory disorder, and the idea is the same. We use the sapphire rod with internal fibers and obtain two signals from two channels. Specially resolved like it's shown here. I should mention that the dimension of the diameter of such a probe is near 5 millimeter. And we also used a rat in vivo, and we induced an ischemia in its leg by fixed the artery. And we detected the signal during 25 minutes and detect that the average attenuation coefficient decreases during this time. So we can conclude that this probe can be used for intraoperative control of ischemia, circulatory disorder, and it can find its advantage. It can find its applications in such fields like reconstructive medicine, plastic surgery, and so on. We understand that further studies are in order, like the study of quantitative thresholds, precision, and sensitivity of such method. And the final part concerns diagnostic scalpel. This scalpel is fabricated from a sapphire ribbon with rather thin width, less than 2 millimeter, with internal capillary channels where optical fibers are placed. We have shown by numerical simulations that we can focus the light using only in the geometry of such a scalpel. We can focus the light right at the end of the blade in a rather small spot. The diameter of the spot is 1.8 millimeter. So we eliminate the tissue just where the blade is moving, when the blade is moving, and we can detect, for example, pathological tissues, tumor or tumor border. This scalpel mainly works based on fluorescence spectroscopy, but the diffuse reflected signal is also very strong. And in this talk, I want to pay your attention only on the diffuse reflectance. Please see the blue lines here. And there are two examples of basal cell carcinoma ex vivo, which we studied just after the excision. And we see that these examples have invasion region, the tumor invasion. And right in the region of tumor invasion, we notice the rapid increase of the diffuse reflectance. In this sample and in this sample also. That's why we concluded that we can detect the invasion using the diagnostic scalpel, which is very important for surgeons. And the last example of the diagnostic scalpel concerns the brain glioma. We used the red brain glioma, the model 109.8, which has very smooth borders, the invasion region also. And also in the brain, we can detect the invasion by the increase of the diffuse reflectance intensity. So instead of conclusion, I want to say only the same thing, that combination of specially resolved diffuse reflected optical response with sapphire-shaped crystals opens new possibility in diagnostics and treatment. And thank you for attention. Please, questions.
Irina, thank you for the very interesting presentation. Let's send the speaker. Any questions?
Thank you for the interesting talk. As I understand, in the frequency domain technique, you have 110 megahertz modulation, and the source detector separation is about 4 and 8 millimeter. Yes?
We have 4 channels.
And
you can see the distance between channels.
Up to 6 millimeter. So this is, as I remember, it's quite low, and their phase shift will be very small for this modulation frequency, I assume. And it would be difficult to differentiate mu-A and mu-S prime from this measurement.
Yes, but right? We didn't use the phase parameter. We use AC and DC parameters here, and that's why we try to reconstruct mu-S and mu.
That's why you return to continuous.
Yes, yes, okay. We returned. We start from the frequency modulation and understand that it is arranged for our purpose.
Yeah.
Yeah, and we go back to the stable state mode.
Okay, this will work on several centimeters.
Yeah, okay, maybe for some purposes we will use it in the future.
Okay, thank you.
Some more questions? If not, I think we may thank the speaker once again and move further. And our next speaker is Andrei Lugovtsov from Lomonosov Moscow State University. The talk lays a method for measuring the microbiological and microcirculation parameters of blood after therapeutic plasmapheresis.
Thank you very much, Ivan, for introducing me. Hello for everyone in Russia. I'm say hello for Chinese participants online. And first of all, I would like to thank the organizer of the conference for inviting me to give an invited talk here. And this work was performed in collaboration our laboratory, physics laboratory, with medical doctors laboratory. from Medical Research and Educational Center of our university. And this slide demonstrates my co-authors, the head of our lab, Professor Priezhev, and the head of extracorporeal therapeutic department of medical research center of our university, Dr. Kalinin, his colleagues Yuri Gurfinkel, Maria Rudnitskaya, and Irina Tauson, and our PhD student, Peter Yermolinsky. Here's the outline of my presentation. First of all, I will say about some characteristics of blood microreology and microcirculation that I will measure in my work. And then we will discuss some techniques which we used very briefly, because some of these techniques were discussed during the previous talks on this session, especially Professor Priezhev in his talk discussed some of these techniques. And after that I will show you my result about alteration of microcirculation and microreological properties of red blood cells and platelets after the therapeutic plasmapheresis procedure. That procedure of therapeutic plasmapheresis, this is a method of extracorporeal hemocorrection aimed to changing of qualitative and quantitative composition of blood, correction of the physico-chemical state of the blood, remove pathogenic substances from the bloodstream, and to normalize. The aim of this is to normalize the immune response. Simply saying, therapeutic plasmapheresis, this is filtering the blood plasma. That means that blood plasma constitution during this procedure is changing. It may affect on blood microbiological properties, especially on interaction of cells from which blood constitutes, the aggregation properties and other properties. And this is very interesting because there is no information till time about the effect of the plasmapheresis on microbiological properties and as a result to microcirculation. There are several phenomena that affect blood microbiology, that affect blood flow, red blood cell deformability, platelet segregation, and red blood cells aggregation. These slides demonstrate the deformability of red blood cells. Here the red blood cells that was deformed in shear flow. This is the ability of red blood cells to change their shape, passing through capillaries with diameter less or equal with diameter of red blood cells. Very important phenomena, and as you understand, its effect of blood viscosity as a consequence, its effect of blood flow. Next phenomena, platelet segregation and red blood cell segregation. The effect can be the same when the cells aggregate, then can change. They may change viscosity of the blood, and it can lead to change in the blood microcirculation. So it is important to measure these properties and to understand how they changed. I will skip this slide to save some time for Chinese colleagues. And the aim of my talk, the aim of my work was to assess microcirculation, deformability, and aggregation properties of the cells drawn from patients before and after therapeutic plasmapheresis for pregnant women. In our work, we used laser optical techniques, methods for studying the aggregation properties of the cells. And the basic of this is to register the scattering or signal or transmission signal from blood samples. So the experimental schematic layout of experiment is very simple. The initial beam illuminates red blood cell suspension or blood, or whole blood, or platelets rich plasma, and then we register transmission or scattering signal, somehow to analyzing this signal and make conclusion about aggregation properties of the cells. Laser diffractometry technique, which we used in our study to estimate the deformability properties of red blood cells, is described on this slide. The essence of this technique is to obtain and subsequent analysis of diffraction pattern obtained from highly diluted suspension of red blood cells. We can see the examples of typical diffraction pattern which we can obtain during laser diffractometry technique. Here is a diffraction pattern obtained from not deformable red blood cells, and here's the diffraction pattern which was obtained from the suspension of red blood cells in shear flow, because all these cells were deformed in the suspension. By analyzing this diffraction pattern, we can calculate the dependence of deformability index on shear flow. That means on deformability, on the shear rate that arises, or on shear stress that arises in the experimental microchannel that we use in this experiment. Another techniques, as I said, was a technique in which we registered the scattering signal, laser scattering aggregometry technique. We used disposable plastic microcuvettes. In this microchamber, we put the whole blood samples. Then we destroy all the red blood cells aggregates in this sample by rotation of microstirrer inside this chamber, and then we immediately stop the microstirrer rotation, and the process of spontaneous aggregation starts in this sample. And we can register the time dependence of forward scattering light intensity of the signal, and the dependence is demonstrated from the right side of this slide. You can see that intensity is increasing within the time, and by analyzing this aggregation kinetics, we can calculate several parameters. First parameter, this is the aggregation index, which is characterized a number of red blood cells aggregated in the first 10 seconds of the process of spontaneous aggregation. And we can calculate the characteristic time of red blood cells aggregation, or aggregation rate. Another parameter which we measured with laser aggregometry technique, critical shear stress. This is technique is very similar, like I discussed on the previous slide, but in this case we registered disaggregation kinetic, and we can obtain disaggregation kinetic signal, the intensity of light backward scattered from suspension of whole blood, from whole blood. This is a dependence on time. In the microchannel, we create the shear stress that is decreased monotonously with time, and the intensity signal is changed. By analyzing of disaggregation kinetics, we can find the equilibrium point. In this point, the aggregation and disaggregation process of red blood cells is equal between each other, and this point is characterized the hydrodynamical strengths of aggregates. We call it critical shear stress of red blood cells aggregates, and we can estimate this. So this is the first two techniques that we use to estimate deformability and aggregation in vitro. Another in vitro technique, this is a technique which is based on registration of light transmission. We illuminate platelet-rich plasma with laser beam and then register the transmission of laser light. And we can estimate the platelet segregation. But before the experiment start, we destroy all the platelets aggregate with magnetic stirrer in the experimental cuvette and add adenosine diphosphate to activate platelets. After that, the process spontaneous aggregation is starting, and we can register a transmission signal, and the typical curve or aggregation kinetics of platelets is presented on this slide. This is light transmission signal, dependence on time, and analyzing this, we can calculate aggregation degree. This is a maximum value of the light transmission. And the next parameter, the aggregation rate. This is the maximum rate of light, the value of light transition through the sample of platelets-rich plasma. This is the slope of this curve. So another technique which was discussed very in details in the topic of Professor Priejev, vital digital capillaroscopy allows us to estimate different parameters of microcirculation in vivo, in particular to measure the capillary blood flow velocity. So this is the examples of capillary blood flow obtained in vivo from normal or healthy individuals and individuals with different diseases. Professor Priejev demonstrated this slide already, and we can see that the capillary blood flow velocity is different in control case and in case of different diseases. So it is interesting for us how the procedure of therapeutic plasmapheresis will affect the capillary blood flow. This is briefly to describe the design of our study. We used 10 samples that was obtained from pregnant patients in the age starting from 25 up to 38 years old. And we used 2 points of blood samples before the first plasmapheresis procedure and after the final plasmapheresis procedure. There are seven plasmapheresis procedure were treated with patients, and let me demonstrate our results. There is no statistical significant difference in deformability index of patients before and after plasmapheresis procedure, but we demonstrated that aggregation index of red blood cells decreases statistically significantly after the plasmapheresis procedure, and the characteristic time of aggregates formation increases accordingly. Increases after the plasmapheresis procedure. Critical shear stress that is characterized hydrodynamical strength of red blood cells aggregates statistically significantly reduces in case, reduces after the plasmapheresis. The similar results were obtained for platelets aggregation, but in this case, for platelets, the aggregation rate of platelets after plasmapheresis increases, and aggregation degree increases as well in that case. And the last result about capillary blood velocity, which was measured with digital capillaroscopy, we can see that after the 7 therapeutic plasmapheresis procedures, the capillary blood flow velocity statistically significantly increases in comparison with the parameters of velocity before the plasmapheresis procedures. And please let me conclude that the therapeutic plasmapheresis results to erythrocyte aggregation decreases, platelets aggregation increases, deformability on the average slightly decreases, but always remains in the range of healthy people in the physiological range. And microcirculation after this procedure improves. I would like to acknowledge our financial support, Photonics and Quantum Technology, Digital Medicine in Moscow University School, and grant with this number. And I would like to thank you for your attention. Thank you very much for Включи.
Hello? Yeah. Thank you for the presentation, and I have 2 questions. The first is methodological question. When you are measuring platelets aggregation, do you have to separate the red blood cells from blood before this measurement?
Yes, because for measuring the platelets aggregation, we obtain platelet-rich plasma without red blood cells by centrifugation of blood. So
And
just
do
we
you
measure it separately.
Do you estimate the quality of this separation? So
Well,
do you have a guarantee that there is no red blood cells in there? And
Well,
if yes, can they contribute to the signal?
This is a typical procedure to obtain platelet-rich plasma, so we just believe that we used this sample without red blood cells. I think so.
Okay, thank you. And the second question is regarding the last slide, which you have demonstrated with the increase in blood flow velocity as a result of plasmapheresis. Have you done the control experiment with the same manipulation but without plasmapheresis? So because perhaps this is a reaction to stress, not plasmapheresis itself. Maybe this is a reaction of an organism on insertion of needle into vein and so on and so on.
I see. Mikhail, very interesting and very important comment. So we have not make these control measurements because this is our preliminary results, which we obtained just only for the blood of 10 patients. So just it will be interesting to check. Yes, thank you.
Great.
Thank you for your
Thank
comment.
you. Thank you.
And here is another question.
Thank you. I would like to ask you, if possible, to comment in more detail. In more detail, this experiment with nails capillaries. Can you show this slide? I cannot catch completely what is shown here. What does this red circle mean?
So, as I understand, you're asking about the vital digital capillaroscopy, and we can measure blood flow velocity. So, first of all, we can visualize and analyze the images of capillaries of blood moving in these capillaries. In eponychium, here in nail bed, you can see this area where we can visualize the capillaries with help of microscope.
And may I look at the previous slide?
You mean this one? You mean
no?
No.
With
This one.
This one.
Yes.
what does this circle, red circle, mean? It is a size of capillary or what?
Yes, this is just capillaries in Elbert's. Yes, this is our
It's a
capillary.
loop, not circle, yeah?
Yes.
It's a terminal loop of the capillary.
Terminal. This is a venous part. No, not venous. Arterial part, and this is venous part, and this blood goes to the
Oh, I see.
Yes. And in the normal case, this looks like this. In the case of different diseases, it can be changed, alterate, and the shape of these capillaries can be changed. So
thank
it's
you very
very
much.
interesting to estimate, and we can measure the blood velocity.
Okay, let's thank the speaker one more time. And we now move to our delayed talk by Sivan Lee.
Sivan Lee.
Yep. Are you with us, Sivan? Sivan, are you here? We can't hear you. Please check your microphone. We see your presentation.
Can you see my screen?
Yes.
Can you see my screen?
Mm-hmm.
Okay, thank
we
you.
see your screen. Please switch to a full screen mode.
Okay, we've already full the screen.
Mm-hmm. Yes, we hear and we
Shall we
see.
start?
You may start.
Okay, thank you.
Good afternoon, everyone. I'm Zhang Yiqing from Professor Li Siwen's research group at the China Pharmaceutical University. And today, I have the honor of representing Professor Li, who is unable to be here due to his teaching task. Professor Li sent his regards and apology for not being here in person. Now, I will give a presentation titled Multimodal Collaborative Tumor Precision Therapy Based on Phototherapy. I will present the following aspects. As we all know, a tumor is the first killer to human health, and the traditional cancer treatment methods include surgery, radiotherapy, chemotherapy, physical therapy, and biological therapy. Our research project is based on physical therapy, especially phototherapy. Phototherapy includes photodynamical therapy and photothermal therapy. We rely on phototherapeutic group to convert light energy into chemical energy and heat energy to kill tumors. It mainly involves three influence factors: photosynthesizers, light, and molecular oxygen. The advantages of phototherapy mainly include little tumor, good selectivity, good applicability, and cooperation with surgery to improve the curative effect. In our research, we have overcome two challenges. Firstly, cascade targeting to achieving accurate delivery. This delivery method can rely on non-invasive, no drug resistance, and high efficacy. Secondly, cascade therapy to improve curative effect, aiming to achieving multimodal cancer treatment with phototherapy activated cascade reactions in the body. Combining therapy, and chemical therapies. Phototherapy can be combined with various treatment methods. When combined with immune therapy, it can increase tumor antigens, explode, and, and promote immune response, thereby achieving local treatment and systemic treatment. Combined immediate treatment with long-term treatment. Phototherapy includes photothermal therapy and photodynamic therapy. They can both combine with biomedical therapy to kill tumors. Next, we will plan to introduce our research project and the results we have obtained. As mentioned earlier, staging is an essential factor of phototherapy and a crucial reactant involved in photodynamic action. The concentration of oxygen plays a significant role in the effect of phototherapy. Our first study was focused on oxygen supplement photodynamic therapy. In this study, the hemoglobin and photosynthesizer CE6 was chemically coupled to form protein itself, loaded with the enzyme enhancer, forming the nano-drug delivery system. It combined oxygen supplemented photodynamic with the enzymes, proposing a strategy for enhanced OS to increase the effectiveness of oxygen to kill tumor cells. It can observe that the cells have the ability to supplement oxygen and increase oxygen saturation. Properly, we used the matrix MMP2 responsive peptide as a linker. When the nanodrug delivery system enters the tumor tissue and encounters MMP2, it degrades and releases the drug. We found that after photodynamic therapy, the expression of two targets, SLC7A11 and SLC3A2, increased, which related to anti sensitivity, explaining the enhancement between oxygen supplement, photodynamic, and MDIs. Based on this, we conducted other study on oxygen supplement photodynamic therapy. We know that tumor tissue exhibit hypoxia after photodynamic therapy. Our research aimed to provide secondary oxygen supply to the tumor tissue after enhancing photodynamic therapy. Improving hypoxia and analysis why the improving hypoxia could enhance the immune microenvironment. Especially, we use the catalyst and CE6 to form protein cells, wrapping with red blood cell vesicles outside of the cells to create the drug delivery system. A red blood cell, the surface vesicles of the drug delivery system. It can transport oxygen to tumor to produce primary oxygen supplement. And near-infrared flight stimulation. C6 produced RS broke vesicles, exposed catalyst ensued, and produced a cascade of oxygen supplement. The secondary oxygen supplement can be observed from the results, and this effect could be observed at the cell level. Moreover, after the cascade oxygen supplement, the immune microenvironment could be effectively improved, achieving the combination of photodynamic therapy and immune therapy. Next, we explored how to compare phototherapy with other treatment methods. The first study involves the use of micropage exosomes fused with liposomes, loaded with photosynthesis TMZ and siRNA-14 therapy. It creates a nano-drug delivery system for the treatment of brain tumor, which not only has the biological function of macrophage exosomes, which can cross the blood-brain barrier through the interaction with ICAM1, but also has the strong drug-loading ability of liposomes. And for thermal stimulation, ICAM1 is upregulated, promoting the drug delivery system reach the brain tumor site. Analysis of tumor patient sample confirms the inhibition of the targets in tumor stem cells. Additionally, photothermal therapy enhances the entry of small nanoparticles into the tumor tissue and effectively suppress the development of tumor in mice. In the second stage of compound therapy, we use the platelet vesicles fused with liposomes, carrying FAC and GX to create nano-drug delivery system. It can target the damaged inner wall of blood vessels because of its platelet vesicles. After photothermal treatment, small nanoparticles can be recruited to reach the tumor site to play a repeated effect. In addition, happy thermal can improve the catalytic activity of GX, accelerate the catalytic rate of FAC, and play the role of compound therapy. From the treatment results, it can be seen that the nano-drug loading system can generate abundant reactive oxygen species during the treatment process, effectively gut tumors and inhibit tumor development. For the third combination therapy, we constructed the drug delivery system called ELP, which use the microphage exosome fused with liposomes to target tumor tissues with high expression of with CAM1. After photothermal stimulation, the expression of VCAM1 was upregulated, allowing more ELP could be accumulated in tumor tissues, achieving cascade driver delivery. Meanwhile, it can provide a CRB killing effect, and it can improve tumor microenvironment. A promising therapeutic effect is shown here. In our first combination therapy, we constructed a biomimetic cascade delivery system by assuming photothermal sensitivity lip cells and outer membrane outguarding of tumor tissue and T cells within tumor reactivity. The fused drug delivery system not only retains the biology characters of the outer membrane vesicles, but also enhances the drug loading ability. The immunogenicity of the outer membrane vesicle and the photothermal effect can observed increase the infiltration of the T cells, and comparing with PD-1 antibody. The silencing of CD38 can effectively improve the T cell cytotoxicity. This study comparing photothermal therapy with immunotherapy showed excellent inhibitory effects on tumor. Furthermore, on our research on cascade reactions, we designed a tumor vaccine based on cascade immunity. They used the red blood cell vesicles to wrap iPSC protein. The aged red blood cell membrane can be retained in the spleen, and the iPSC protein acts as a broad antigen. This nano-vaccine effectively delivers antigens to the spleen, achieving a dual effect of tumor treatment and prevention. We sincerely thank the following folks for their support and the members of our research group. My presentation is over. Thank you.
Thank you for the interesting presentation. Let's thank the speaker. Are there any questions? Maybe in online? If not, I have one tiny question. Is it possible to measure, maybe in percent, how effectiveness of the treatment increases when you combine a number of therapies?
Sorry.
Should I repeat the question?
Thank you for your question.
So
I'm sorry. There is no answer on this issue at present, and we will pay attention to this question in the later research.
Okay. In this case, I think we will thank the speaker one more time. And our session is ended for today. See you tomorrow.
Thank you.